Today we are going to discuss nitrogen fixation. So first of all, let's talk about nitrogen. Molecular nitrogen is major component of the atmosphere. Approximately there is 80% of nitrogen in the atmosphere. The element nitrogen is essential part of many chemical compounds such as proteins and nucleic acids which are basis of all life forms. However, nitrogen molecule cannot be used directly by biological systems to build the chemicals required for the growth and reproduction. Before its incorporation into a living system, nitrogen molecule must first be combined with the elemental hydrogen. This process of reduction of nitrogen molecule, commonly referred to nitrogen fixation, may be accomplished chemically or biologically. Need of nitrogen fixation. Nitrogen is essential component of DNA, RNA which are present in human beings and proteins, the building block of life. Although the majority of the air we breathe is nitrogen, most living organisms are unable to use nitrogen as it exists in the atmosphere. All we know that the nitrogen is about 80% in our atmosphere. Due to inertness of nitrogen molecule and high bond dissociation energy of nitrogen molecule, so all most of the living organisms cannot use nitrogen molecule from the atmosphere. Now, what is nitrogen fixation? Nitrogen fixation is a process that converts the atmospheric nitrogen to useful compounds of nitrogen. The conversion of nitrogen molecule to reacting form of nitrogen is called nitrogen fixation. Nitrogen fixation is done naturally by microbes. Humans can also do nitrogen fixation in industries as fertilizers production. How nitrogen gets fixed? When nitrogen combined with oxygen or hydrogen. The very first step of all fertilizer production is based on the reaction discovered in early 20th century by two scientists, Haber and Bosch. What is nitrogen fixation? Nitrogen fixation, any natural or industrial process that causes free nitrogen and two molecule, which is relatively inert gas plentiful in air, to combine chemically with other elements to form more reactive nitrogen compounds such as ammonia, nitrate or nitrites. Nitrogen is fixed or combined in nature as nitric oxide by lighting in and UV rays, but more significant amount of nitrogen are fixed as ammonia nitrites by and nitrates by soil microorganisms. When there is lightning in atmosphere, the nitrogen present in atmosphere dissociates as simple nitrogen and molecule, and that molecule combine with hydrogen and oxygen present in atmosphere, so it can form nitrogen compounds which can used by plants or living organisms on the earth. More than 90% of all nitrogen fixations is affected by them. Two kinds of nitrogen fixing microorganisms are recognized: free living bacteria which are non-symbiotic, including the cyanobacteria or blue-green algae. The symbiotic nitrogen fixing bacteria invade the root hairs of host plant where they multiply and stimulate the formation of root nodules, enlargement of plant cells and bacteria in intimate association. Within the nodules, the bacteria convert the free nitrogen to ammonia, which the host plant utilizes for its development. The root hairs of host plant absorb nitrogen and they can form nitrogen compounds from that nitrogen, and host plants utilize these compounds for their growth. Within the nodules, the bacteria convert free nitrogen to ammonia, which the host plant utilizes for its development. To ensure sufficient nodules formation and optimum growth of legumes, seeds are usually inoculated with commercial cultures of appropriate rhizobium species, especially in soil poor or lacking in the required bacterium. Nitrogenous materials have long been used in agriculture as fertilizers, and in the course of 19th century, the importance of fixed nitrogen to growing plant was increasingly understood. Accordingly, ammonia released in making coke from coal was recovered as utilized as a fertilizer, as were deposits of sodium nitrate from chile. Whenever intensive agriculture was practiced, there arose a demand of nitrogen compound to supplement the natural supply of soil. At the same time, the increasing quantity of chile salt, saltpeter used to make gunpowder led to worldwide search for natural deposits of the nitrogen compounds. Mechanism of nitrogen fixation. The general chemical reaction for the fixation of nitrogen is identical for both the chemical and biological processes. There is a molecule of nitrogen and three molecules of hydrogen. The sum amount of energy is required to break the triple bond present between nitrogen molecules, and we get two molecules of ammonia. The triple bond of nitrogen be broken and the three atoms of hydrogen must be added to each of the nitrogen atom. Living organisms use energy derived from oxidation of carbohydrates to reduce molecular nitrogen to ammonia. The chemical process of nitrogen fixation involves burning of fossil fuel to obtain electrons, hydrogen atoms, and energy needed to reduce molecular nitrogen. This is the reaction for nitrogen fixation. Now, the nitrogen fixation involves non-biological nitrogen fixation and biological nitrogen fixation. Then, the biological nitrogen fixation can be accompanied by two processes. These are non-symbiotic and symbiotic processes. Non-biological nitrogen fixation. In this process, microorganisms do not take place. This non-biological nitrogen fixation found in rainy season, which we can say acid rain. The nitrogen present in atmosphere react with oxygen during lightning to form nitric oxide. The lightning energy can break the nitrogen molecule, and it gets reacted with oxygen to form nitric oxide. And this nitric oxide also react with oxygen to form nitrogen dioxide, nitrogen peroxide, and this may further react to form nitric acid, and as further we get ammonium nitrite in last step. Biological nitrogen fixation. Fixation of atmospheric nitrogen into nitrogenous salts with the help of microorganisms. In non-biological nitrogen fixation, there is no need of microorganisms, and in biological nitrogen fixation, there is need of microorganisms. These are further of two types: symbiotic and non-symbiotic. Non-symbiotic, the fixation carried out by free living organisms, aerobic, anaerobic, and blue-green algae. These are some bacteria given which are used in non-symbiotic processes. Also, these are yeast and Pluria, blue-green algae, and some bacteria which are used in non-symbiotic processes. Now, symbiotic processes: fixation of free nitrogen by microorganisms in soil, symbiotically inside the plant. In non-symbiotic, these are free organisms present in air or atmosphere, and in symbiotic, these are fixing microorganisms present in soil or inside the plants. Now, nitrogen fixation process are also split in vivo and in vitro. In vivo processes are within a living organisms and in vitro processes are involved in artificial environment outside the living organisms, as we can say production of nitrogen fixing as urea. Now biological fixation. There are two types of nitrogen fixation bacteria. These are free living bacteria as non symbiotic and the roots. These roots are symbiotic which fix 70% of nitrogen. Now in vitro nitrogen fixation. In vitro is Latin word which means within the glass. A procedure performed in vitro is performed not in living organisms but in a controlled environment such as test tube or a petri dish. In vitro process, this process is performed in as industrial or labs because of test conditions may not correspond to the condition inside the organisms. This may lead to the result that do not correspond to the situation that arises in living organisms. Consequently, such experimental results performed in vitro are contradicted in vivo. In vitro experiments are performed in model in vivo experiments. Simply, we can say in vitro nitrogen fixation can be done in laboratories or industrial use, but these may also contradict the results from in vivo nitrogen fixation. The enormous energy of lightning breaks nitrogen molecule apart and enables the nitrogen atom to combine with oxygen form nitrogen oxide. Nitrogen oxide dissolves in rain forming nitrates. Nitrates are carried to the ground within the rain. Now these plants use nitrates for their growth. Now industrial fixation. During the first decade of the 20th century, intensive research efforts culminated in the development of several chemical nitrogen fixation processes. The first, the arc process. Air is passed through an electric arc. About 1% nitric oxide is formed, which can be chemically converted to nitrates. By 1902, electric generators were in use as Niagara Falls, and by this venture failed commercially because it was costly and inherently inefficient in its use of energy. The cyanamide process. Reaction of nitrogen with calcium carbide at high temperatures to form calcium cyanamide, which hydrolyzed to ammonia and urea. This was utilized on a large scale by several countries before and during World War One, but it too was energy intensive. The Haber-Bosch process directly synthesizes ammonia from nitrogen and hydrogen and was the most economical hydrogen fixation process. The arc process and cyanamide processes are very costly and energy consuming processes. So Haber-Bosch process is economically nitrogen fixation process. Importance of in vitro nitrogen fixation. It provides a model for in vitro nitrogen system and to employ molybdenum. It provides insight into a development of useful catalyst for industrial fixation of nitrogen. Limitations of in vitro nitrogen fixation. It is not a continuous process. After fixing one nitrogen molecule to two ammonia molecules, the reaction stops. The reaction stops when a metal is completely oxidized as there is no more electron supply, but in vivo the reaction goes on because there is an enzyme nitrogenase which assists the continuous supply of oxygen and hence the electrons. In vitro process stops but in vivo process doesn't stop. Now in vivo nitrogen fixation. In vivo nitrogen fixation accompanied in between the plants or organisms as rhizobium bacteria in nodules present in roots of plant. They convert nitrogen to nitrogen compounds, and these nitrogen compounds are used in plants for their growth. In vivo nitrogen fixation. In vivo processes are processes which occur or made to occur within a living organisms. Certain symbiotic or free living bacteria are able to fix nitrogen into useful compounds. Symbiotic bacteria fixes dinitrogen in association with plants. For example, the bacterium rhizobium, which is associated with the nodules of the roots of leguminous plants. A symbiotic bacteria are free living bacteria which fixes atmospheric nitrogen. For example, azotobacter. In biological nitrogen fixation, 2 moles of ammonia are produced from 1 mole of nitrogen gas using 16 molecules of ATP and supply of electrons and protons. Here ATP stands for adenosine triphosphate. This is a reaction for preparation of ammonia. The bacteria contain the enzyme called nitrogenase, which is responsible for nitrogen fixation. Nitrogenases are the complexes of iron sulfur protein and molybdenum iron sulfur proteins whose activity couples ATP hydrolysis to electron transfer from ferredoxin for carrying out a reduction of nitrogen to ammonia. Now, iron protein. It is protein with molecular weight near about 66 kd and contains a single 4 iron and 4 sulfur cluster, also known as nitrogenase reductase. It passes electrons from electron source like ferredoxin and pass them to p cluster. This is structure for iron protein, in which there are 4 iron and 4 sulfur molecules. Now there is p cluster. It mediates electron transfer to the activate cofactor site during catalyst. It has chemical composition of 8 iron and 7 sulfur molecules. The p cluster is believed to assist the reaction by transfer of electrons. It accepts electrons from the iron protein and transfer these electrons to iron molybdenum cofactor, resulting activation and reduction of nitrogen. During the 2-electron redox cycle, the p cluster undergoes significant structure change between two oxidation states, pn cluster and pox cluster. Now molybdenum iron cofactor. It consists of two cuboidal fragments with molecular weight of 220 kd. One contains 4 iron atoms and the other 3 iron atoms and 1 molybdenum atom. Molybdenum is octahedrally coordinated at the corners of heteroquinone structure and is coordinated to histidine and tetraanionic chelating homocrate ligand. The vital feature of iron molybdenum sulfur cluster is activate site situated in the center. Now biological nitrogen fixation. It is extreme energy consuming conversion because of stability of the nitrogen molecule. Produces fixed nitrogen which can be directly estimated into nitrogen containing biomolecules. This is the reaction for biological nitrogen fixation. What is biological nitrogen fixation? Conversion of atmospheric nitrogen to ammonium ion. Under natural conditions, nitrogen fixation is the main pathway by which new available nitrogen enter terrestrial ecosystem. Non-symbiotic nitrogen fixation. These are the names of the bacteria which are responsible for non-symbiotic nitrogen fixation. Nitrates are essential for plants. These nitrate ions, which may be present in soil during acid rain or some other symbiotic processes, the roots of plants can take nitrates from soil, and these nitrates are used in the growth of plant. Nitrates are recycled with microbes. The plants or animals which are died on the earth. These are decomposed by microbes or decomposers, and they decomposes dead bodies, and nitrogen compounds also liberated from these dead bodies, which are mixed in the soil, and these are also get under many reactions, and these are formed, and nitrate ions are formed, which are also used for the growth of plants. This is also the example of nitrogen fixation, as the lightning energy dissociate nitrogen molecule, and these forms acid rain, which are converted into nitrates, which are and present in soil. There are also biological fixation, soil organic nitrogen, and these all nitrates are used in the growth of plants. Atmospheric nitrogen fixation. These are fixed by electrical storms. Lighting provides sufficient energy to split the nitrogen atoms of nitrogen gas, forming oxides of nitrogen